So today our topic is going to be recombinant DNA technology and stuff. But to understand that, first you need to know what DNA is. So DNA is biological information. That's all it is. And if it's biological, you can expect it to be in each and every cell of your body. And to be more specific, it'll be in the nucleus of the cell. So you have DNA, which is formed into chromosomes, and these chromosomes have specific alleles on them and genes. These genes, they code for everything from your blood group to the type of hormones you produce. They make you... What happens is, in certain people with genetic disorders, they might be suffering from something like phenylketonuria or any other disorder, or maybe even a disease like diabetes, and their body may not be producing enough hormones or enough proteins, and this causes various problems in their body. So traditionally, over the ages, we have gotten these hormones from places like cows or pigs or other human bodies. But you can guess that there are lots of complications when we use these. It'll be very expensive. And it might cause allergic reactions in people because especially if it comes from cows and pigs, people are not used to proteins from them, and it might cause allergic reactions, which might give them worse disorders. And with such little to go around, and so many people suffering from diseases, there's just too little. So in some way, we want to produce more of these biological products. And how are we going to produce them? We need something that is cheap and something that is easy to prepare. So we want something which has the best of both worlds. We want human proteins and human hormones, but we want them cheap and we want them easy. So another place we have already started using this thing of two separate worlds is in hybridization. To understand this, it's good if we take the example of hybridization. What happens in hybridization is we take two plants and cross them over to get better plants with like really good colors and flowers and new things in them. Another place we have seen this being used since like time immemorial is alloys. For example, bronze is an alloy made from copper and tin, and bronze happens to be stronger from either of them. So it is stronger than copper, it is stronger than tin. So in the same way, we want something which is cheap and which produces human hormones. So enter recombinant DNA technology. So here we are going to take, this was developed by some pioneers of genetic engineering back then, Stanley Cohen and Herbert Boyer, who got together in a conference, and later in 1973 they developed a method of having bacterial cells produce hormones for human beings. Now this technique could be used to produce anything practically, any biological products. So what their technique basically involved was take a bacterial cell or any other microbe, anything that is cheap and easy to grow. Now bacteria have their own genetic information, and sometimes they also have an extra loop of genetic information called a plasmid. Now this duplicates separately. It can be easily transferred to other bacteria without too much of a fuss, and it's pretty easy to work with. Now, what they did was they figured out that something called restriction endonucleases. Now, when you hear the ending of nucleases, aces, you can guess that it is an enzyme. The ACE tells you it's an enzyme. And this enzyme helps to cut open genetic material. So we can take the plasmid out and cut it anywhere we want. And in the same way, we can take a human gene and scientists figure out which part. For example, let us say this part codes for insulin, which is the product we want to make in, let us say, this example. So using those restriction endonucleases, we can cut out this much and we can cut open this plasmid. So we get a cut open plasmid and we get just the part of the chromosome which codes for insulin. And if you put them together, we get something which is called a recombinant DNA, because it has DNA from both sources. It is called a recombinant DNA. Now, this recombinant DNA is taken from a bacteria which is carefully chosen to be antibiotic resistant. Now, I encourage you to pause the video and think about why it should be antibiotic resistant. So, did you think about it? So basically now we have a recombinant DNA ready to go back into a new bacteria. So let's draw another bacteria here. But it has its own genetic information already, and we are going to shock this bacterial cell using electricity or chemicals to make small transient holes, like they're short-lived. So the plasmid goes in, and you can see that, as I said, it should be antibiotic resistant, so it'll have a little antibiotic resistance gene which will continue to come here, and these holes will close up after some time. And after this transgenic, because now it should be called a genetically modified organism, will now be put in a culture medium. And this culture medium, along with its nutrients and all, will also have an antibiotic. And this antibiotic will be the same one to which it has that resistance gene. Now, this is very important because during the process of taking up a plasmid, some bacteria might not take it up no matter how much electricity we use, so they won't have that plasmid. And we want to get rid of them because we don't want them competing with this bacteria for nutrition or anything. So by adding this antibiotic and having this antibiotic resistance gene, these bacteria will be wiped out, and only our plasmid containing bacteria will survive. And another thing about bacteria is they flourish very quickly, they reproduce very quickly, so we'll see lots of other bacteria along with the plasmid because it also duplicates when the bacteria is duplicating. So this whole thing will be full of bacteria, and when the scientists think it's time and there are enough bacteria, they will be taken out of their culture medium, and all of the bacteria will be processed using enzymes which will digest, which will remove the cell walls and leave us only with the products which our plasmid has coded for, which in this case will be insulin. So these cell walls will be removed, and all we'll have will be insulin. This insulin is, of course, impure, and it will need to be purified, but effectively we have used an organism which doesn't have any gene for insulin, and we have formed insulin from it using a very simple but still very amazing technique recombinant DNA technology.